Estamos sistematicamente todo mundo que por acaso. Yeah. Consta. Então estamos ao vivo no YouTube aqui.
It's almost.
Okay, so I guess we can start on time because we're all set. Gente, por acaso. Tá pessoal no lugar aí. Estamos ao vivo no YouTube aqui. Tem um feedback. Eu tenho feedback de som, talvez de você, Mari.
Sim, peraí.
Se você está assistindo no YouTube, tá dando ou se muta, ou muta o YouTube, na verdade. Né?
Já mudei o YouTube.
Tá, beleza.
Parou, né?
Parou. Bom dia, gente. Nós somos o Brisa. Obrigado pela casa cheia hoje. Esse é o nosso quarto simpósio anual. Eu vou... Enfim, a gente tem convidados internacionais, então vou mudar para inglês em alguns segundos. Mas, enfim, duas boas-vindas a todo mundo em português aqui e tal. Se alguém tiver perguntas em português, depois a gente está super... enfim, disposto a falar língua. A gente deve ter um tempinho depois do simpósio para trocar uma ideia sobre a nossa chamada. Eu vou falar um pouquinho sobre ela agora, mas a gente vai ter tempo depois para discutir isso. Eu vou fazer essa introdução, a gente vai ter os 3 palestrantes e depois a gente vai ter tempo para as perguntas para os palestrantes e depois sobre a chamada. So, yeah, this is about it for Português. A gente não está gravando, né? So good morning, everybody. Thank you for coming. Thank you, Clarice and Gro, our speakers. Benjamin should be joining shortly. He had an emergency issue and he should be joining in 20 minutes or something. We're Brisa. We're the Brazilian Reproducibility Initiative preclinical initiative. In the Brazilian Reproducibility Initiative in preclinical systematic reviews and meta-analyses. And I'll be briefly presenting the group. This symposium is also happening because we have a call from members. We want you to know about Brisa in order to see whether you might want to join. So let me share my screen here and start. So Brisa is a collaboration of around 40 basic biomedical researchers. Some of us have a little bit more experience in that, but many of them actually started out with no experience at all, which is part of the fun. People who come from various fields of biomedical science, particularly animal science, and are interested in learning more and performing systematic reviews. We started out as an offshoot of the Brazilian Reproducibility Initiative, which is a large replication project, which is coming to an end. Actually, we should have results in a couple months. But we started out just before the COVID pandemic. So like people in the project were stranded for a while without a lab. And we're just trying to figure out what to do with so many people and so little to do experimentally. Somebody, I actually don't know who, proposed this idea. Yeah, let's do some systematic reviews. And we kind of like bought into the idea. We found people who had some experience in that. We had some informal training in order to start some projects. And we soon realized there was kind of a niche to be filled and that was actually a good idea. So actually training biomedical researchers in performing systematic reviews helps them both being dependent in this kind of methodology. And I think it actually helps them being a better experimental researcher as well, because you kind of like realize some things. And there's also a niche in terms of catalyzing collaborative projects. I mean, systematic reviews usually need a lot of hands to happen. So actually getting people together who are interested in a similar topic could be of use. And we realized those two goals actually complement each other quite well, because actually the training involves getting involved in something. We had around 20 people from the first informal gathering. We then launched the first call for members in 2022, in which we selected 30 more people, I guess. We put up an online training course, part recorded, part synchronous. The recorded part is actually all online in this link. And we selected 30 people. And with those 30 people and the former members, we were able to start, 4 other systematic reviews projects. So we actually are working on 6 projects at the moment. We hope to finish at least half of them this year. One is having a manuscript written already. 2 or 3 should end more or less in the middle of the year. So we have room for new projects, for people to pitch new projects. And so we're actually here today for 2 reasons. One is actually to discuss interesting aspects of systematic reviews in different fields, in vitro studies with Gro, in animal studies with Benjamin. Clarice will talk a little bit about statistical issues that come up in basic science, in which papers are not structured in the same way as the clinical ones usually. The other reason is actually to advertise that we have an open call until next week, March 11th, so that's next Monday. This open call is open to anybody who has any experience in biomedical research. You don't have to have experience in systematic reviews properly, although that is okay as well. So we're actually looking for researchers from all levels, from undergrad to senior, with a background in basic biomedical research. Our focus is on basic research. I mean, so like if you're fully clinical, that might not be exactly our niche. And people who want to learn more about systematic reviews and engage with us in collaborative projects for hands-on learning. So you can apply in this link, bit.ly/Inscrição Brisa 2024. You have to send a motivation letter and optionally an idea for a systematic review. You don't have to have your own idea. You can work in other people's ideas. Actually, most people will have to, because we cannot do 30 projects at a time. We will look at the applicants, select around 30. We have 60 applications at the moment, so it's going to be tight. We should notify you by the end of March if you're accepted, and then our online course is expected to run from April 9 to July 16, with lectures and synchronous activities that happen on Wednesday afternoons. Sorry for those who are busy on Wednesdays, but it's hard to find a time that fits everybody, so it's better to state it up front. We have a basic systematic review and meta-analysis course. There's topics in Portuguese. And after the course ends, we'll have a round of for people to pitch projects in August. We'll discuss project proposals, and collectively we'll try to select the most promising ones and the most popular ones among our group to develop in Brisa in groups of whatever, 5, 6, 8, 10 people, depending on how many hands you need for a project over the course of a year or two, which is usually what takes us to complete a systematic review. And if this sounds interesting to you, we'll be happy to welcome you on board. So just apply until next Monday, and we might be talking over this year. And this is it. We can now move to our speakers. We will have 3 short talks around 15 minutes each, and then take questions for everybody at the end of the symposium. And then if there's time after 10:30, probably we can take questions on the Brisa call if anybody's interested to know more about how the group works. And this is it, I guess. So I guess it's my pleasure to welcome Gro Matheson from the Norwegian Scientific Committee for Food and Environment, who's going to be talking about how to evaluate validity in vitro studies, particularly cell line studies. She's currently the head of an effort to standardize some ways to evaluate validity. So thank you a lot for being here, Gro, and the floor is yours. I'll just stop sharing here.
Thank you, and thank you so much for inviting me and giving me the opportunity to present this project. So I think I will share my screen like this. Does it work for you now? Because I'm struggling a bit with
yeah, we can see you.
Zoom, but it's okay. Perfect. So this is the project I'm going to present for you. It's called the Validity Assessment Tools for In Vitro Studies. And as you can see, I work at the Norwegian Scientific Committee for Food and Environment. And this is the Norwegian risk assessment body, which is governmental. And so in my work, I do chemical risk assessments. And as part of the chemical risk assessments, we do the hazard assessment. And then we very often do systematic reviews as part of the hazard process. So this was the background for initiating this project. And the project is funded by the European Commission through the Horizon Europe, and it's a part of a European partnership, which is called European Partnership for the Assessment of Risk from Chemicals. And the abbreviation is PARC, and you can see it's quite a large partnership. So It has about 200 partners, includes 3 European institutes, so, you know, both Chemical Agency, Food Safety Agency, and also the Environmental Agency. And it will last for 7 years, and we are in our 2nd year now. And so what I will present for you then is our work on creating tools for validity assessment of in vitro studies. And the background for this, as I said, is that we are doing systematic reviews as part of the hazard assessment, and we need it to be based on the best available evidence. And then in the process of both identifying the data and also when we turn the data into evidence, we need the processes to be comprehensive, objective, reproducible, and transparent. And then therefore we are using the systematic review methods in our risk assessments. So for chemical risk assessment, we are turning from using animal testing to other types of data, as I'm sure you know. And in vitro studies are one of the types of the new data that we are going to need to be able to use as evidence. And we don't have the tools we need yet to use this data. So it's not, for now, possible for us to use in vitro data as evidence in our chemical risk assessments. So what we need, for instance, are tools for validity assessment. So this is what we are creating. And we are actually creating 2 tools. One tool for evaluation of the internal validity of an in vitro study, and one tool for the evaluation of the external validity of an in vitro studies. And so these tools are then called Invites In and Invites X. And then, so very briefly, Invites In will then be used for every study to evaluate if there could be alternative explanations for the result of the study. So that will then be the risk of bias, and then the internal validity. And the external validity tool will be used to evaluate whether the cause and effect connection reported actually can be generalized to humans, and then how it can be done like that. So we have been working mainly on creating Invites In for now. It's the easiest thing to start with because there are so many internal validity or risk of bias tools out there already. So we had kind of a starting point. Whereas for the external validity, there are very limited number of tools and much harder to really define what it's all about. So we are just in the beginning of the Invites X, but we are not there that we have all Invites In created already, but we have come a good way. So I will present for you now our methods and so how we are doing it. So then just to illustrate the systematic review process for hazard assessment, then as I guess you all know, then all the first 5 boxes will be part of the systematic review process. And then for the hazard assessment, then we have the hazard conclusion and so on, which is the last box. And the tools we are creating, they will be applied here in these 2 boxes to evaluate the risk of bias, and also then for evaluation of the certainty in the body of the evidence. And so we are creating these tools specifically for cell culture studies. So we say it's a tool for in vitro studies, but in order to be able to really make sure that it works for one kind of studies, we are specifically designing it for cell culture studies. And we had spent a lot of time to design the method of how we are going to do this. And so it was very important for us to make sure that we follow a rigorous approach, that the whole process is systematic and transparent and open and available for everyone interested. And we also involve different experts on in vitro models and also on systematic review methods through the different studies we are doing to create the tools. So we have a project team and also an advisory board. And then in addition, for every study we do, we include additional experts that are outside the project team and the advisory board. And so you can see here, we will do 4 studies actually to create the tool. And these are the 4 studies we are going to do. So 3 studies are done to create a tool, and then the 4th study will be the testing to see that it really works in practice for the users. So the 3 studies we are doing to create the tools are then the first one to discover factors that could be of relevance for introduction of bias to the in vitro studies. And then in the second study, we want to rank these factors according to importance. So then to rank and select factors that are of importance for assessing the potential for bias in the in vitro studies. And then the third study will be the creation of the tool. And to make sure that we are open and transparent and follow rigorous methods, we created a protocol that we published before we started. And so this is the reference you can see here. And when the tool is created, we're going to test how well it works for the users. And then we have a separate protocol that describes the whole testing procedure, which is the other reference you can see here. And so just to give you a bit more insight into these studies. So in the first study then, when we want to discover the factors that are of relevance for introduction of bias to in vitro studies, we are both using literature sources and focus groups with in vitro experts. And then in the second study, when we are ranking these factors according to importance, we are doing a Delphi study. And for this study, we have experts in both in vitro studies and systematic review methods that are helping us to do this. And then in the third study, when we prepare the draft tool, we will invite in vitro experts to help us to make sure that we are describing the guidance documents and phrasing everything according to the best terminology to make it understandable for all readers. It's a bit tricky in this field that the systematic review methods use another terminology than actually the chemical risk assessments do. So it's a process to really find the best terminology to use. And as you can see in the user testing study, we will also then involve these experts on in vitro studies. And the first 2 studies, then we hope to make sure that we really aim to be comprehensive, that we capture all kinds of factors that are relevant. And then in the 2 studies, we really aim to ensure that it's as user-friendly as possible so that the tool actually can be used and without too much struggle. So to give you a bit more detail about the first study, this study has been done. So we are about to send the manuscript now. And just to give you an overview then of the sources, I said we used the literature sources. And so we started with the literature and we chose the sources you can see here. So it's 5 bias assessment tools and it's 1 study sensitivity tool. And it's also 1 systematic review, which where they created an item bank, so a bank with factors that could be relevant for introduction of bias. And so we extracted everything of relevance from these sources, and then we did the focus group study. So we had, for the focus group study, we had 20 participants from 8 countries. All participated in 2 meetings, and they were divided into 3 groups, so in 6 meetings in total. And through these groups, we aim to identify the factors that are not yet captured into the literature, because none of these tools have gone through a very rigid procedure to include factors that are relevant for in vitro studies. So that's why we had in vitro experts here to make sure that we captured additional factors that are of relevance for in vitro studies. So this is the process that we went through. We started by abstracting the factors that could be relevant. And then we interpreted these factors into internal validity issues that were addressed, and then we converted these factors into statements, which we called items. And then all these items were mapped onto different bias domains. So they were grouped according to the type of bias. And then finally, we removed the duplicates. And again, to make sure that we are a bit rigid in what we are doing, 2 persons did the first 3 steps independently, and then it was followed by discussion and reconciliation. And for the mapping onto the bias domains, 1 person did the mapping and another one checked. So then just to show you the outcome. So the number of bias items that we discovered from the literature from these 7 sources in total were 311. And then from the focus group, we discovered an additional 114 items. So the focus group turned out to be the single source that gave the highest number of bias items. And this is how they were mapped onto the bias domains. So for instance, 55 items were mapped as being analysis bias, 23 to be attrition bias, whereas, for instance, only 3 items were mapped to be timing of study termination bias. And as you can see, we ended up with 405 unique bias items. And so these items are then included in what we call an item bank. And this is only actually a file with all the items. And so everything here will be published to be open and transparent. And the reference you can see is here. The manuscript is for clearance now at 2 US institutions, and then we hope to submit it within the next 3 or 4 weeks. So the second study, which was the Delphi study, is the study where we want to have experts ranking the factors according to importance. And so it's a 2-round Delphi study, and it's followed then by a workshop, which will be a guided discussion. And in the first Delphi round, we included all the 405 items. And in the second round, we have included 237 items. And the second round is yet ongoing. So it will end by the end of this month. And then we will have the workshops with the guided discussions. And so you can see here we have in total 17 participants from 7 countries, and they all have systematic review and in vitro model expertise. This is just to illustrate how the Delphi study is designed. So what we are doing is that we are having all the 405, at least in the first round, we had all the 405 items and presented for the participants. Here are 2, just to give you an example. And then we have asked the participants to rate the agreement for each item with the following statement. And the statement is then, this item should be considered when assessing the potential for bias in an in vitro study. And then they have this 5-point Likert scale for rating it. And so when the first round were done, we looked to see if we had some agreement, and then we say that an item is included in invites in when 70% of the experts moderately agree or strongly agree with the statement. And it's excluded from the tool if 70% of the experts moderately disagree or strongly disagree with the statement. And the statement then is that this item should be considered when assessing the potential for bias in an in vitro study. So as you can see from this slide, then we moved from 405 to 237. So the number between here is the number where we received agreement in the first round. And this is just to give you an idea about how the user testing study will be done. So it's a very long, detailed protocol, but this is just to show you that we will look into 4 things in the user testing study. So we will, to test the performance of invites in, we will look into consistency. And then with consistency, we mean the extent to which results of the application of the tools are the same for different users. So this is what is called iterator reliability. And we will look into the assessor workload, both then the amount of time it takes to apply the tool and also the cognitive burden or intuitiveness of the participants who test the tool, if it's too hard to use or requires too much effort and so on. And then we will look into the accuracy. So this includes also 2 things. It's one of them is the precision of the results of application of invites in between the user results and a reference evaluation. And then the second is the potential for systematic differences between the results of the users and the reference evaluation. And then the last thing we will look into is the user experience. And then we will collect qualitative feedback about the process of applying invites in, how it was, if it was too time consuming, impossible to understand the guidance documents and so on, just to really try to get some feedback there. And so I've just included this one. Volunteers will be needed. This is what we have been doing for all our studies. We have tried to find persons that find this interesting and would like to participate in the study. So I think for the Delphi study that's ongoing now, I think we have at least 1 Brisa participant. So if anyone are interested, you're more than welcome to participate in the user testing study if you have some in vitro model expertise. And then this is just the conclusion then that invites in. It's a validity assessment tool for in vitro studies, and it's for internal validity. And as I've tried to show you, the process for creating the tool is quite rigorous, and it's systematic, transparent, and we started out with literature, but we have also included expert knowledge in every step. And we have this 4-step process of first finding the relevant factors, then the important factors, and then to draft the tool and test usability. We publish the protocols in advance, and we have experts involved also all the time, also to make sure that we anchor this work throughout the community and also to ensure the usability. What I didn't say is that for each study, we will publish all the materials. So for instance, when we submit the first study, when we created the item bank, we will publish transcripts from all the focus group meetings and so on, to make sure that everything is open and available. And we think that this tool is important for turning in vitro data into evidence. So it will be used to look into the potential for systematic error. It's important to be able to evaluate both the certainty in the evidence, but also to describe the uncertainty in the evidence, and to make sure that when we do our systematic reviews, that they are comprehensive, objective, reproducible, and transparent throughout the process. So this was it, and I just included these links. So in case anyone would like to have more information, we have a web page for this project that you can find under this link. Then you can see the park symbol and click on that one, and you will find the web page for this project. Thank you.
Thank you, bro. That was fascinating and a lot of work, I guess. If you can just send the links in the chat. I started to, but so just people would know how to join if they want to collaborate. But that was very interesting. So we have been joined by Benjamin Ineichen from Zurich, so who's our next speaker. So you just dropped in, but yeah, it's kind of your turn. So Benjamin will talk more about animal studies and how to assess their validity. So go for it.
Yes, hello everyone, and sorry for joining late with an emergency meeting coming in, but happy to be here. I'm happy to talk about validity of animal studies, and as you guessed, I will talk about systematic review, mostly translation. So I'm a research group leader at the University of Zurich in Switzerland at the Center for Reproducible Science. And I'm a medical doctor, so I'm very interested in translation. So when you develop drugs in animal models, when you model certain human diseases in animals and you translate them across the species barrier to humans for eventual human application. And my team and myself are working on assessing translation on a very large scale, and I'm happy to share my presentation. Can you hear me well, by the way? Yes, thanks.
Yep.
So I'm also a neuroscientist. I have a PhD in neuroscience, so I'm particularly interested in the translation in neuroscience, and our research is mostly funded by the Swiss National Science Foundation. Oh, is it the wrong screen you're seeing?
It's the wrong screen. Yeah. We see the speaker screen.
Yes. Now?
Yeah. Good.
Yes. Okay. Thank you for pointing this out. So the audience, may I briefly ask, what's the main background of the audience? Is it mostly scientists?
Yeah, mostly I would say people involved in biomedical research, basic biomedical research.
I see.
I don't.
Okay.
I mean, I don't know about everybody, but
Of course.
many of them will be undergrad and graduate students, but mostly from. I mean, we might have some clinical people as well, but mostly basic bio.
Right. Yeah. No, makes sense. Thanks, Olavo. That's very helpful. So the translation of drugs or how drugs develop is probably to the largest part of the audience is relatively clear that you test drugs first in animals and then you translate them. And so in preclinical testing, this could also involve in vitro testing, and then you advance them to early human trials, and then later stage human trials and eventual market approval. The details are not important. We are interested in the translation of drugs from animal models to humans and eventual market approval. And this process of developing novel drugs or any kind of treatments for any diseases is very, very inefficient. So this is a relatively old paper already. It's 20 years old, but I can tell you the rates haven't changed. So out of 100 drugs which seem promising in animals, roughly 90% drop out in clinical development due to a variety of reasons, toxicity, inefficacy. But this shows that this process is extremely inefficient, and many animal experiments are conducted without actual gain for clinical translation. And I would argue that one of the biggest arguments for still conducting animal experiments is to inform human health. It's not the only reason, but I think one of the big reasons. So this process is very inefficient, and we try to understand which factors govern the translation from animals to humans. And if we understood these factors better, one could potentially improve or enhance that whole drug development process. And our main methodology, you might have guessed, is the systematic review. I think most of you already have quite some idea what a systematic review is. It's a rigorous review it's a reproducible and rigorous summary, basically, of all the literature on a given question. And I won't go into every step here, but basically you have a comprehensive literature search in different databases, and then you go through all these papers you find and decide for each of these papers, is it relevant to answer my question? And you extract predefined data, and finally you combine all these data together to draw a conclusion on the question you asked initially. And the problem now, as you might already have guessed, is conducting a systematic review on the translational rate. So all the drugs which have been tested for neurological diseases in animal models, and how many of them actually made it to clinical trials or eventual market approval. That is hundreds of thousands of studies, even for a narrow field like neuroscience, not considering oncology, cancer research, immunological research. So there is hundreds of thousands of papers out there, and there is more and more papers being published every day. So you can see from this graph that last year alone, almost 400,000 new animal studies have been published last year alone, and this is further to rise in the future. In addition, all these data are very unstructured. So all the data about which drug have been tested in which type of animal model and so on is just in this hidden in this huge pile of papers. I mean, it is written in the papers, but it is very. I assume you have read many research papers, and it's very non-harmonized, the data. Thanks. Sorry. Yes, so that's a problem. We try to assess the translation, but that's the barrier. We have a lot of data to work with. And the idea is that we do a comprehensive assessment of the translational rates for neurological and psychiatric diseases. So which therapies have been tested in neurological diseases and how many of them reached clinical applications. And then once we have this huge data frame of all the drugs which have been tested in all models and with all the outcomes, we can employ statistical modeling to potentially see predictors which could then be associated with successful translation. And all these data will be made available in a data warehouse. So researchers can access all these data without reading all these papers themselves. And so what we will do is we will identify all the relevant literature, or this is what we're currently doing. So basically all papers which have tested any kind of therapy in an animal model of a neurological or psychiatric disease. Then, since these searches are usually very broad, so very sensitive but not specific, we'll have a lot of false positive papers in there. We will sort the library for relevance, fetch all the full texts of these hundreds of thousands of records, extract relevant data like which drug has been tested, which dosage, for which, in which species, and so on. And do the same thing for the clinical trials. So which drug has been tested and is employed in the clinical setting for neurological diseases. And finally, showcase all these data in a warehouse. And again, we will not be able to do this manually. So we will use natural language processing approaches like large language models. So I guess every one of you has now heard of ChatGPT. So we will not just ask ChatGPT, but we will use the underlying models, GPT-4 or other. There's also lots of other open source language models, which we will train to specifically extract all this data from the papers. And I won't go too much into detail, but one thing we are currently working on, I would like to share with you is the extraction of data. So there will be hundreds of thousands of studies, as I said a couple of times already. And how will we extract data from all these hundreds of thousands of studies? And yeah, we'll skip that. But we have, so two master students in my group, they developed this data mining toolbox in R, the R programming language. We work a lot with R. And this toolbox basically takes a PDF, so you can hand 1 or 10 or 1,000 PDFs, how many you like, to this tool in R, and this tool will convert the PDF to text and then parse the text into different sections. Then you would have the introduction and the method section and the result section, and then it employs a library of terms to these different paper sections and extracts specific information from these sections, like which species has been used, like rats or mice. And then it stores all this data in a data frame for every paper you hand it to this tool. And it can extract animal species and model, as well as sex, like whether female or male animals have been used. It can extract outcome measures, and also study quality in air quotes, risk of bias. I think most of you have heard of that. So whether the study is at risk of bias, so whether it was blinded or randomized, which is relatively rare for animal studies. And the tool performs relatively good, depending on the item, between 80 and 100% accuracy or F1 score, depending on what you're interested in. And so this tool works for relatively easy extraction tasks, like if you want to know, has this study used rats or mice or rabbits or whatever. But there is more complex extraction tasks as well, like, for example, which drug have been used in a certain study. And so the problem with drugs is there is 10,000s of different drug names and synonyms and drug codes from different pharmacological companies. This is a much more complex extraction task if you want to know which drug has been tested, and there is actually no comprehensive library available. So we use these large language models, among others GPT-4. So we fine-tune these models to do them what we want specifically. And a PhD student of mine, yes, Simona Doneva, a PhD student in my lab, she's a computer scientist, and she developed one of these models. I mean, she didn't develop a model, but she used the model, but fine-tuned it to extract drugs and diseases from scientific abstracts or clinical trial registries in that case. But one could also use that for preclinical, for animal studies, for example, potentially also in vitro studies. And the model performs relatively good. So we manually annotated a corpus of around 900 studies. So we basically just highlighted manually, or I mean in the computer program, but like we went over all these records manually and highlighted the drugs and highlighted the disease, and then basically gave this whole corpus to the model and said, well, this is the drug and this is the disease, and now tell me for the rest of these 10,000 publications, what was the drug and what was the disease. And these models learn very well. So they can extract the drug and the condition, as you can see in this red square, with F1 scores of around 80%. F1 score is a measure of how well these models perform. And so around 80% is also human performance. So a human doesn't have 100% because humans also make errors in these tasks. So they reach human performance. And so this is a proof of principle that these models can be used to automate certain systematic review tasks. And these models will now also be expanded to extract specific information from animal studies. And before I finish my presentation, I would like to show you where this all is going. So as I said in the beginning, we would like to have this data warehouse. And we have a pilot set up already with 99 studies. So it is just the beginning, obviously, 99 studies. But just so you get an idea where this is heading. And we call this Anemone, like animal models in neuroscience. And then you can go to the graphs and statistics. You can see these 99 studies, how many animals it used, what was the risk of bias. Then you can select a therapy, for example, fingolimod. This is a drug. And then most studies used mice, for example, and this drug has been translated for multiple sclerosis, for example. So it is clinically used. 5 studies in total were used for fingolimod. You could also select all drugs which have not yet entered the clinical trial. So again, you see how many animals in total have been used, which species, again, mostly mice, which animal models, no clinical translation, again. And then you see here all the drugs which have not yet entered clinical trials. And then there's the data warehouse. Every single row is one study with metadata, like which year study has been published, how many animals have been used, and what is the drug, what drug has been tested, what was the outcome, and some risk of bias items. Was the study randomized? Was it blinded? And a couple of other things. And again, this is just for 99 studies now currently, but this will relatively soon have 100,000 of entries with all the data mined. So what is the goal of this research? One of the primary goals is to enhance animal welfare, to make this whole drug development more efficient and thus use less animals. But at the same time, obviously develop novel, contribute to developing novel drugs conceptually. And also the tools we develop can help to automate the systematic review of animal studies, but potentially also in vitro studies or other preclinical domains like in silico. Yes, the take-home message to close with is this project is still work in progress. We have made some progress, but I've showed you basically how we approach a big data systematic review with some automated steps along the way to enhance the translation. And with this, I thank my team and my funding, and I thank all you for your attention.
Thank you. That's also a lot of work, and I guess we can talk more about how to make it wider and more available after the talks. So we'll have the last talk now, which is Brisa from Brisa itself, who'll be talking about some statistical issues coming into the analysis of data from basic science in meta-analysis, and we then move to discussion. Again, we can take questions in the chat. If anybody has questions ready for the first two speakers, you can leave them in the chat. Otherwise, you can also ask them live after the three talks are finished. Brisa?
Yeah, thank you. Thank you for having me here again. And yeah, so today I'll be talking about the nested data structures that happen in basic research, and I hope to make this understandable for those who don't have a lot of experience in meta-analysis or statistics in general, but I'm happy to take questions as well. So just a brief overview. I hope most of you have a general idea of what a meta-analysis is, but just to get everyone on the same page. A meta-analysis is a statistical summary of several studies. So here, for example, we would have the effect of protein synthesis inhibitors injected into the prefrontal cortex of mice and rats on the acquisition or consolidation of peer memories. The scientific component here is not so important, just take this as the effect of the drug on behavior in mice and rats. Each square here represents a study with its confidence interval. And then the diamond represents the meta-analytical summary effect of these 6 studies. And so if it goes into this direction, you would say that this intervention is improving memory. And if it goes in this direction, you would say that it's disrupting memory. And so the summary conclusions from these 6 studies would suggest that these drugs are disrupting memory formation or acquisition or consolidation. And this is based on this publication, that are all of the examples that are going to be coming from this publication here. I'm sorry, can you see the bottom of my slides, or is this the
there's some
showing?
Yeah, we can see the first line of the reference, but the second seems to be missing. So yeah, there's
Is this now?
Yeah, this is better.
Okay. Yeah, sorry. Okay, so yeah, I'll be taking all the examples from this publication, but don't focus too much on the scientific component. I hope to explain more the main issues here. So just like any statistical analysis, meta-analysis also need to have some assumptions made, and one of which is that the observed effects are independent of each other. So in a basic research preclinical setting, if you have a treatment being given into the cage for 3 animals, you would not it would not be appropriate to take each animal as independent of each other, as an independent observation. The correct here in the primary experiment would be to take the cage as the experimental unit. But the same issue can be seen if you take multiple experiments from a single paper. How independent of each other are they? And then, of course, if you have a large pile of papers, could you see maybe some characteristics that make some papers more similar to each other than they are to the rest? So here, if you had these yellow papers having some kind of similarity, that makes them not independent of each other, and then makes them more different from the blue papers, for example. So just to illustrate this. this is not so much of an issue in clinical meta-analysis because papers from clinical studies have typically fewer outcomes than preclinical studies, so that multiplicity of results from each paper is not so much of an issue. They also have fewer groups per trial, so when they come to a clinical trial, of course, the drug is already, well, that has a lot of evidence behind it to select which group should work best. And they also have more clearly defined primary outcomes and comparisons of interest, so this could also help the person doing the systematic review and meta-analysis to filter through what is really the most relevant from each paper. And in the case multiple cohorts or subgroups are included in the clinical studies, this is typically also easier to determine their independence of each other. So take, for example, if you read a paper that was published now in 2024, and they conducted a study between 2019 and 2022, and they include a group of children and a group of adults. This is very clear that these are not the same people being accompanied throughout time. But in preclinical studies, this distinction, if you take an animal with 1 month and 3 months and 6 months, you are not as sure that they are not the same animal being accompanied throughout time, or if they are different cohorts of animals. So, and if you take preclinical research or biomedical research as a spectrum from very basic discovery research to translational drug, translational component or applied research, the more you go towards the basic field, typically the papers will have more outcomes, more groups being compared, and this issue of the cohorts not being very clearly labeled. So this makes it harder to determine the independence between the effects within each paper, within each preclinical paper, or even between some papers. And why is this a problem? So if this assumption of independence is not met, and then you are still treating these observations as independent, if they are positively correlated, the error is underestimated and the precision of the summary effect is overestimated. So the width of that diamond that I showed in the bottom of the meta-analysis, this will be artificially made narrower, so you would have a lot more confidence than you should in the summary effect estimate that you are taking from this meta-analysis. We proposed the use of multi-level meta-analysis to kind of address this issue in preclinical meta-research. Of course, we are not making up this methodology, but just proposing a way to apply it. And the difference here is when you take the fixed effect model, which is the most basic model that you can do a meta-analysis with, it is only considering the sampling error within each experiment, so only between the animals that are composing this graph in the paper. If you go to a random effects meta-analysis model that is the most commonly used in preclinical research, it's also including the sampling error of the experiments or between the experiments. So you have here 2 levels of variability that are being included. And what we are calling multi-level is when it has more than 2 levels. And then you could also be adding more layers of sampling errors, for example, within the papers or within research groups. These are the examples that we used in this publication. So here we had identified 313 experiments or comparisons, and up to 18 experiments per paper, and up to 58 experiments per research group. Of course, most of them have only 1 or 2, but still there is this curve here of a lot of experiments that are coming from the same source. And briefly, you may wonder how we are defining research groups. We are applying this methodology that was developed by a former colleague of mine, Thiago, who so this methodology is based on co-authorship graphs. So we get all the authors for all the publications that we selected to include in the meta-analysis. We see which papers they are co-authoring, and the more papers they are co-authors in, the stronger their connection is in this graph. And then we can see these clusters being formed. Each dot here is an author then. Some of them only share 1 paper, so it will be just this easy small cluster, 5 authors with 5 in 1 paper. But Some of them have more connections and also connect through multiple papers. So this author here, you could see they are connected with this side, it's probably one paper, this side is probably another paper, which then connects this group through another publication through this other author. And so these are all through the mathematical properties of this co-authorship graphs or network graphs. They get assigned to this one cluster. We get the number for this cluster. This is called the modularity class. And then we get each paper that we selected for our meta-analysis to be assigned this identifier of which research group the paper is coming from. So going to the results, this is a lot. So in our publication, we looked into different data sets, but as I said, I don't want to focus too much on the scientific content or the distinction here. I want to focus only on this reactivation data set. What's important to know here is that this is one of the effects there's most contentious in the literature of fear memory, and we identified for this data set 145 experiments, and we tested the 2-level random effects model, the 3-level considering the variability within articles, and the 3-level within groups. So we had 56 papers for these 145 experiments and 28 groups for the same number of experiments. We can see here that the effect size is quite consistent between these different models. But when we look at the heterogeneity here on the right-hand side, we see there's a large heterogeneity. Here, this is 89%. But then when we look into the 3-level models, also a very relevant proportion of this heterogeneity is explained by each of these levels, by the article or by the group. And this is a very high heterogeneity in general by design. So we were looking into protocols that had a lot of variation between them. So just to give some examples here, we had various possible sites of injection, the different models for conditioning the animal to acquire this memory, and the different characteristics of the animal. So yes, exploring the source of heterogeneity was also a goal of our study. And we could do that through meta-regression. So looking for the association of each variable, each protocol variable, which can be seen here as each line in this graph. And then the different data sets are represented by each column. But let's focus first on this corner here, just to see what this graph is all about. Each tile, each brick here has the color representing the statistical significance. So the darker the color, the more significant it is. And the number inside is the R-squared value. So the higher the value, the more of the variability between the experiments can be explained by this protocol variable. And if we focus again on the reactivation data set, which is this column right here, we see conditioning type, number of shocks, and re-exposure duration appearing as significant variables in the random effects model. And if we go now to the article level, the 3-level model with article, the numbers already change a little bit. And then if we go to the model with the group, we see a very big reduction, especially here in re-exposure duration, suggesting then that the group was a confounder here in this analysis. And from this 19% variability that was explained here, actually this was being confounded by the effect of group, and group actually explained a large proportion of the variability between these experiments that's not necessarily related to the re-exposure duration, to this protocol variable specific. Of course, these are a lot of protocol variables, and they can be also confounders for each other. So we also did some multivariable multi-level models. So we looked for the best multivariable model that explained the variability between the experiments within each of these data sets. So for the reactivation, if we focus here again, we see the number of shocks, re-exposure duration, and injection site as the 3 variables that appear in the best multivariable model when we are taking the random effects model of calculating meta-analysis. But then again, when we look to the 3-level by group model here, the proportion of variability explained by each of these variables is actually very much lower, suggesting again that the group is the most relevant, likely the most relevant characteristic of these experiments that would explain why the effect goes one direction or the other, that would explain the variability between these effects. And yeah, so as a conclusion, I want to say that the nesting is likely a common issue in preclinical research. This was definitely a common issue in this very small field that we got here on peer memory and behavior, but this is likely to be a more broader problem, and we should try to account for it in preclinical meta-analysis. We are suggesting the use of multi-level models, but this is not the only solution. It also comes with some limitations, and in general, just like any meta-analysis, multi-level meta-analysis are also limited by the reliability of the literature itself. So if the effects that are being published in the literature are not as reliable, there is no statistical method to correct for that. But the multi-level model is still a good option to account for the nesting issue. And we also suggest in this publication that investigating this heterogeneity of the effects in the literature could be complemented by high-powered confirmatory replication studies. So we suggest that this, for example, these 3 variables that were found here in the multivariable, multi-level models could be included in an experiment, in a confirmatory experiment that has very low risk of bias and very high power, statistical power for detecting these effects, to see if an experiment could actually validate the meta-analytical findings. And we could take these results again into the meta-research and then go back to refining the meta-analytical methods to inform future studies, testing new drugs and new interventions. So yeah, this is what I had, I think. So thank you. Oh yeah, these slides are available in this link. I will also post this in the chat. Thank you.
Thank you. So, well, we heard from all 3 speakers now. Thanks for keeping on time, by the way. And we're open for questions. You can raise your hand. You can just burst in talking. You can use the chat. I think anything works. If no one else starts, I will. So actually, this is a question both for Gro and for Benjamin. I mean, you both talked about tool development, either a risk of bias scale or a warehouse to store data. That said, I mean, there's a lot of people working on tools in a lot of parts of the world. I mean, Bro, you mentioned you found already some existing tools, and there's probably some people working on that as we speak. MC3Rs has just come up with a River, which is it's a reporting guideline. There's no risk of bias, but I mean, that kind of goes together. Same for Benjamin, and I have seen this. I mean, people trying to compile large pieces of the literature in organized fashion in many areas. I get the idea that it works better if everybody is on the same page and working on the same tool. How do you deal with like this multiplicity of tools arising in both of your efforts?
So if I should start, so there is some, there's a lot of tools for evaluation of the risk bias, but not a lot of tools available that are designed specifically for in vitro studies. And the tools that are available, they don't have any rigorous approach behind the design of the tool. So that's why we are creating this tool, to have one comprehensive tool that are designed in a rigorous approach and involving people in the different parts of the community. So in addition to, I mentioned we have this project team, but we also have an advisory board, and in our advisory board we have several persons that are responsible for the risk-based tools that have been created for human studies and animal studies and so on, really to make sure that we can have their advice, and to make sure that also that we are not duplicating any ongoing work. And they are leading a lot of projects around the world, and so they have also then informed all the persons involved what we are doing to try to see if there are any duplicating efforts going on. So seems like thing we are doing is not something that anyone else is actually doing. So, and of course the tool will be, it will have a narrow approach, it will be for in vitro studies. So it won't be for animal studies or so and so, but I think that the way we are creating it and using the methodology with involving all the experts, and for instance then seeing from the focus group study that actually these groups with experts identify more items than the literature itself, so that it's a good way to have this also when you design a tool. So I hope we are not, you know, doing something that's already out there. We have really done our best to try to ensure that.
Yeah, I agree with Ro that there's lots of, and also you all there's lots of tools available. It feels like we are working more from a top-down approach. So we try to solve a problem and develop tools which aim to solve that problem. And since the problem is relatively specific, for the translational question at least, there is not much specific tools available. But you're right, there's lots of groups working bottom up, developing really tools from scratch to extract data. But these tools are not really tailored for specific questions, and they might work well in a very generic context, but if you have really focused questions, they might perform less well.
Adriano?
Yeah, so hello everyone. Thanks for the very nice talks. And I have a question for Gro. So my concern, I mean, the initiative is obviously needed and tries to solve a problem for everyone, and it's a huge effort. So my concern is about the usability, your stage 4, the stage 4 in your study design, because the number of items, original number of items is huge, 400, something like that. And it dropped to 200, I think, after the first round. And to me, it still seems to be high. So what's the expected number of items to have a tool that is useful for the community? What do you think about this?
Yes, so very good question, and I didn't have time to mention that, but a lot of the items that we have included are, you know, partly overlapping, and as you saw, it was like 55 items for analysis bias and so on. And for those who have seen the items, you will see that there are a lot of overlap, but there are slight differences. And because our approach is to make experts outside our project team give us the advice of what are the important and not important one, we have left it all like this. So we have lots of items that are partly overlapping. And so when the Delphi study is done, then we will see which items are considered to be important. And then we will group the items and create signaling questions. That will be the questions that the users will actually use, you know, so they can have a question, you know, saying that was the outcome assessor blinded, for instance, and then a guidance of how to rate this question. And then it could be several items actually that are covered by this question. So what we will do is to try to create the questions in such a way that they will cover more items. So we don't actually for now know how the grouping will be. So we cannot say how many questions it's going to end up with either. But of course, we cannot have, it's not like it's going to be 100 questions to answer for each paper, for instance. That's impossible. And so we are very focused on usability, but we are involving, again, the in vitro experts in the process of turning and grouping the items into questions to make sure that we do it the best way. And I guess also that it could be different kind of groupings and questions for different models of cell cultures and so on. And so to make sure that it works well, experts will be involved and it will be a process. And then, of course, we do the user testing also to get feedback on whether it's too much, whether it's too hard to use or too time consuming and so on. So I want to try to balance it. But it's also very important that we look into all the factors that we need to look into. So it's this balancing part. So that's why we do the step by step and cannot really say for now how many questions or like that, but we have an impression of how much time, for instance, could be used for each paper when you use the tool. So it's not like we can, no one would create a tool where it took 3 hours to rate, for instance, one study. That would be impossible. So it will be narrower, far from the 405 items we started with. Yes, absolutely.
Yeah.
More questions? If not, I'll fill the void. For Benjamin, another further question is how to keep it. How do you keep it living? I mean, I've seen a lot of this living systematic review things when you actually can update, but then the grant is over, postdoc goes away, person finishes a PhD, and this ends up forgotten. And then, I mean, keeping it living is requires effort or just a plan, right? And I have seen a lot of this living review fading on that dimension. So what's your plan to actually keeping this regularly updated?
Thanks, Olavo. It's a very relevant question. As you say, infrastructure changes, people change. Living, we will implement all these tools in a programmatically in an R and Python pipeline, and integrate, or the plan is to integrate it at the servers of our center, like the institute. And so it's not dependent anymore of people. So it will automatically fetch or do the whole filtering and data extraction step. But there's always, I think that's a risk of the project in general, and we have to be aware of that risk.
Alisa?
I have a follow-up question to that, if I may. It's about like reproducibility or translation of this tool. How easy would you say it is for someone to take your methodology and apply to another field, or like how much expertise would that be needed? How much time would that be needed? And yeah.
You mean like individual, for example, data extraction tool, for example?
Yes, if some group in Brisa wants to apply this for some, I don't know, tropical disease, would this be easy, or how much expertise would that take?
That's a good question. I think it depends on the tool. I showed you this R tool, the R Steed. That would be relatively easy. So you would need basic R skills to implement that. The other tools might require a bit more expertise in programming, but still, in the end, the goal is to make them as user-friendly as possible. But I think this won't go without any like affinity for computer stuff. You don't need to be a programmer at all, but you need to write 1 or 2 lines of code, I think. But then you should be able to use them.
Thanks.
Thank you.
Of course, the question sounded heavily interested. I think I know what project you're talking about, so we might get in touch if Adriano.
Yeah, so I have one more question for Grow, which is, I think there is something between cell cultures and in vivo studies, which tissue cultures, organoid cultures, which are the interest in people working with these models is obviously increasing. So have you considered the possibility of using your tool to evaluate experiments using tissue or organoid cultures? Because this is kind of a literature, right?
So, as I said, we are specifically designing the tools for different types of cell culture studies. So that could be, 2-dimensional, 3-dimensional, and so on. So we have detailed it in the protocol exactly which cell culture models that we will include, and that's because we need to be able to power the user testing study, and having too wide range of different models, it wouldn't be possible for us at all to power the study enough, because then we will need several hundred volunteering for the user testing, and it will be quite a lot of work, so it's not realistic to do that. So but in the protocol you will see exactly which types of cell cultures that we will use in the user testing. But we hope, of course, also that the tool will be applicable for all other types of in vitro studies, but then it will need to be tested. So, I guess it would be applicable for other types of studies, but it could be that you need slight changes according to the model and so on, and it needs to be tested. So what we are trying to do is to make something that really works for a specific type of model or specific types of cell models, and then other persons can take it, use our protocol for if they would like for the user testing and so on, to test it for other models to see how it works.
Thanks.
If that answered your question.
Yeah, thank you.
Yeah.
Benjamin, then Anna.
I think Adriano was first.
Yeah, go ahead.
Yeah, so I have a question for Clarissa. So maybe I've missed it, but in animal studies, so I've conducted a couple of meta-analyses on clinical studies, and there you have a problem with heterogeneity. But in animal studies, the heterogeneity between studies is much larger of a concern. Have you considered using Bayesian models as well for meta-analysis, or is this something you can comment on?
I don't think I can comment on that. I don't have any expertise with that. I never applied it. Yeah.
Right.
In our case, at least, the heterogeneity was kind of on purpose, because we were not so much looking for the summary effect, but much more on the regression of like determining which protocols were best for getting to them.
That makes sense. Thanks.
Yeah. Sorry.
Anna?
Okay, so I would like to thank all the speakers for the wonderful talks this morning. And my question is for Clarissa as well. I'd like to know more about the confirmatory replication study you mentioned. Do you think it should also be a multi-lab experiment to avoid further research group, variability, introduce heterogeneity? How could this be achieved? Is this something that you think is feasible in the short, middle term, or what's your take on that?
Yes, I would say yes. Multi-center is always better than single-center studies. I didn't go into much detail about the methodology of what such a confirmatory study could go into, but the idea would be that it would apply the best possible experimental design options for improved internal and external validity and reliability in terms of statistical power as well. And I think in the BIH, we are developing a lot of tools and guidelines for confirmatory multi-center studies. But in terms of feasibility, it requires, I think, specific funding for that. It's not something like a systematic review that you could kind of do as a side project, as a contributor in the lab by students or something, but I think it needs some extra infrastructure support and funding for that. to happen, and it takes a long time as well. Sometimes it requires a lot of efforts in terms of alignment of the different centers, so this takes time. Yeah.
But I think even a single center study would actually help. I mean, it is a methodological step above. I mean, meta-regression is always observational by nature, right? So if you just do it randomized, I think it's a good way to see whether your capturing is actually real, right? We did plan to, in our defense, we did plan to do that. It's just that we took so long to finish the meta-analysis that we had already closed the lab at that time, and we didn't have the physical means to do it anymore. Mari, you raise your hand. No. Anyone else who have something in chat? But Gladys, that's a question for you. Actually, for me, you mentioned the tool for doing the commodity graphs. I mean, shouldn't we have how easy is that to apply these days? I mean, do we have an open source tool that you can just punch in DOIs and get that back, or is it still in the do it in MATLAB, then do it in Gephi, then do it in whatever that we originally developed it?
We have the R code for creating the networks in R, but to get the list of authors, I think this was still implemented in MATLAB. But there's definitely a possibility to do this in R by scraping Web of Science based on the DOIs that you already selected. It's just not what we implemented in this paper, but it's possible to create an R, a short R code for that.
Yeah, I think that's useful. I mean, we should. Philippe is here as well, right? So I mean, that might be something we should be talking about. Anyone else?
Yeah, if I can make another question for Benjamin. Could you comment on what you think is the relevance of internal versus external validity in explaining the translational failures of a given field, perhaps the field you know the most? What are the differential impacts of these kinds of problems?
That's a very, very good question. I can just guess. I think there is no hard evidence on that. My gut feeling would say that the external validity is the much bigger problem. So the internal validity, meaning that many of these studies are low quality, they are not blinded, they're not randomized. And the external validity, meaning that I personally believe that many of these animal models are just not suited to study human diseases. That would be my gut feeling. I've worked for a couple of years with multiple sclerosis animal models, mostly EAE and a couple of others. And I mean, the disease somewhat mimics MS, but it's also so far away. If you look at how the model evolves over time, and multiple sclerosis, for example, is a chronic disease, a lifelong disease. And if you model this in animals, you have one multiple sclerosis relapse, and then the animals recover fully from that. So it's very different. And I feel that scientists sometimes don't really are aware that these models, they just use it because everyone uses them, but actually I think many of them are very poor. So my guess would be that the problem for the translation is really the external validity. Would you agree with that, or what would be your gut feeling?
I think it varies according to the field and according to the model, perhaps, but I think both of the problems impact translational failure.
Gro, would you share that view for in vitro studies as well?
So I think that, for the in vitro studies, the external validity is very important because it's very different if you have cell culture model or even a cell line or something that are not really even normal cells. and then you see something happen, increased production of a protein, increased expression of some kind of genes, mRNA, so on, and then you're going to actually use this to say something about, at least what I do in my work, then to say something about human health effects. And then you really need to know which kind of effect can it be applied to? Is it relevant for which, is it for all? Is it only for a time period during life? You have models for, fetus development, or is it for grown adults? Or so it's so hard. So I think that's really why we started with the tool for internal validity, and why we're spending so much time looking into external validity, because it's much harder, and it's also very, very important, but it's so much less out there to base your work on also. So it's kind of not getting maybe the attention it needs to have also for animal studies and so on.
And maybe following that up with a more conceptual question, I mean, how do you incentivize models to get better? Because, I mean, taking it's a lot of science don't even realize, because, of course, I mean, they get published, right? I mean, you can work in a model that actually has very little to do with a human disease, and that might not help. I mean, you might not even know it, because it doesn't really affect your career very much, unless you're in the very, very top of the translation pyramid, like just trying to get something. I mean, so it matters for pharma that's trying to get something in vivo, but like for the average basic researcher, it might not matter. So like, how do we incentivize this? I mean, do we give prizes for people who work on better models? Do we have a model ranking? I mean, how do you get the basic scientist that actually is not very aware of what's happening on the clinical front to try to look for models with better validity, rather than ones that are more practical or easier, or that he has more experience with?
Yeah, maybe I can make a quick comment. As you already alluded to it in your last sentence, Olavo, what is a better model? In my mind, a better model is someone which leads to better translation, so which predicts translation in humans better. And I think it's an extremely difficult question to answer. For example, there is Lou Gehrig's disease, ALS. There is a genetic model, and 10% of the human cases are also genetic, and the mice have the same genetic mutation. So it's actually quite a good model, because it has the same genetic mutation as the humans. But still, when you test drugs in this animal model, the drugs work in the animal model, but they don't work in the humans. So I wonder whether animal models at all should or can be better than they currently are. Maybe it's a bit provocative to say that, but maybe that's the case.
Yes, I think it would be good if researchers were, more aware of this and looked more into it, and had to actually explain in their publications if it's translatable to some kind of health effect, and the documentation for that. So they need to prepare some basic papers showing or documenting actually how it can be translated. And this is to make sure that the publications can be used, for instance, for risk assessments or other types of assessments. And so, of course, many researchers don't know actually about that. And we see it, and I guess all of you also do that frequently when you do the systematic reviews, that lots of papers or studies end up with very high risk of bias. And it's often because you don't actually know the answer of the signaling questions, because it's too bad reporting. So just, to increase the reporting would make it easier for us to at least be able to assess the risk of bias, and also then if they were informed what we need to know to be able to evaluate the generalizability or the translatability, that would help a lot. But they need to be aware of it also.
More questions? Yeah, so I think maybe a last question for Grow. I mean, how do you see? I mean, we have been doing systematic reviews in cell studies, and to me, like the biggest issue is like you don't know what the experimental unit is. Like, yeah, we did 3 independent experiments. What the hell is independent experiments? I mean, are these different cultures, different pastures of some culture, just different wells in the same place, or we measure something 3 times? And like for most data, I think it's impossible to actually know what has been done in terms of like how many times this has been repeated in what conditions, which leads like to like ridiculously small error bars, colons, the effect sizes of 100 or 50 something, which is probably just like somebody measured the same thing 3 times in a row or something. But it's actually impossible to know. So like how are you looking at that? Because to me, that seems like the biggest gaping issue that doesn't make me trust systematic reviews of cell lines.
So we are creating this tool that can be used to evaluate the internal validity of these studies. And we have tried once to do a systematic review of in vitro studies, and it ended when we came to the risk of bias assessment because we didn't have any tool. And I guess that the answer to your question will be part of the process of what we are discovering on our way of developing these 2 tools. And I guess that most studies, as they are performed and reported today, it will be hard for us to actually use them, but that we will also, when the tools are prepared, create this checklist for researchers to tell them that if a risk assessor or someone else who does a systematic review should be able to include your study, and then we need to, of course, be able to evaluate both the internal and external validity, then this is what you need to report to us for us to be able to use it. And then, if the reporting improves, then I think it will come up, the answer of your question sometimes, but it's not really there yet.
We're hitting 10:30, so that might be any more questions. That might be a good place to end the questions, I suppose. But I mean, we can still take questions on the call, on the Brisa call. So like, I thank all the speakers. If nobody else shows up in 3, 2, 1 with a question. Well, thank you very much. It's been enlightening to talk to you, and even though the lectures were quite different, I think we definitely learned something in terms of how to go forward. Again, we're systematic review collaboration. There's all kinds of holes in basic systematic reviews. There's all kinds of needless work which could be streamlined, and I think this kind of talk is interesting in terms of like how can we do better, and we hope that there's something we would like to advance in Brisa as well. So I guess we can thank the speakers, and if anybody has questions on the Brisa call for new members, either as in Portuguese or in English, the Brisa coordination, which is me, Ana, and Mariano, will be happy to take them as well. If not, I'll just send the link here to the application form. Inscrição Brisa 2024, né? Foi só inscrição Brisa.
Inscrição Brisa 2024, sim.
So maybe I lead them. So thank you so much.
Yes. Yeah. Thanks a lot, bro. Yeah, this is now internal matters but maybe somebody has a question. We're open until March 11, so up to next Monday. Se alguém quiser aplicar para o chamado do Brisa em vigor, por favor, mande sua carta de motivação, ideia de revisão sistemática e informações básicas aí até segunda-feira. O link está ali. A gente está selecionando pessoas nas próximas 2 semanas ali e tal, e entrando em contato no fim de março para começar com vocês em abril. A última chamada foi muito legal para quem entrou assim e tal, e espero que a gente consiga repetir a experiência e tal, começar projetos que façam diferença aí.
I'm also leaving. Thanks for having me.
Yeah, sorry
Goodbye.
for speaking
Thank you very
Portuguese.
much.
But thanks, Benjamin.
Thank you. Talk soon.
Tem uma pergunta
Sim.
aqui de quantas pessoas vão ser selecionadas. Da última vez foram em torno de 30 pessoas, e é mais ou menos esse número que a gente está pretendendo, assim.
que é o que a gente consegue dar conta, né? Assim,
Dá
para
conta.
conseguir acompanhar os projetos, etc. e tal. A gente tem 6 no momento, assim, que é meio que o limite a gente consegue acompanhar. Devem ter 3 ou 4 terminando esse ano, então, assim, a gente conseguiria começar mais ou menos uns 4 projetos no segundo semestre. Foi a nossa conta. E para encaixar gente, etc. e tal, acho que muito mais do que isso é difícil. O curso, de novo, é parcialmente gravado. As aulas estão em bit.ly/curso Brisa. E a gente vai ter aulas síncronas nas quartas à tarde. A maior parte das aulas é gravada, mas, enfim, tem atividades síncronas para você, enfim, para exercícios e para discussões e para coisas mais legais do que simplesmente acompanhar uma aula. Perguntas
E
do
a
fórum?
ideia é que o curso seja um início mesmo, e a participação seja a ideia de produzir projetos com suporte de, enfim, alguém que já participou de alguns outros, tem um pouco mais de experiência para tentar ser algo mais aplicado mesmo, além das aulas formais.
E, de novo, assim, o grupo se propõe, claro, assim, a gente basicamente se propõe a fazer projetos colaborativos para treinar pessoas, assim, então acho que esse é o carro-chefe, né? Mas, assim, a gente se encontra para discutir coisas, para fazer esse tipo de simpósio aqui e tal, então, assim, o grupo tem uma atividade científica limitada, por enquanto, assim, e tal, que a gente não está produzindo muita coisa em termos de pesquisa sobre revisão sistemática, que eu gostaria de fazer, mas tem essa missão de comunicação, de divulgação, de outreach. A gente é parte de alguns comitês internacionais ali e tal, do Prospero for Animals, do Global Evidence Summit, enfim, de algumas coisas ali que a gente está participando, a gente tem uma interação com os fora. Assim, tem participado no grupo, que é aparecer numa reunião mensal e eventualmente assumir algum papel nesse sentido, e tenho participado de projetos, que a gente tenta manter aí uma meia dúzia ativa a todo dado momento. Essa reunião mensal também serve para discutir os projetos. Geralmente os projetos são feitos, os atuais estão sendo feitos em grupos de, sei lá, 5 a 10 pessoas, mais ou menos, o que divide a tarefa e tal. O grosso da tarefa é de extrair informação e, enfim, fazer screening de artigos, etc. E a ideia é trocar expertise também. A gente tem pessoas de diferentes níveis de expertise, de senioridade, de campos diferentes de ciência. Eu acho que isso também nos ajuda a criar um ambiente de aprendizado, né? Para mais sobre o Brisa, aí dá para olhar o nosso site também, produtividade.bio.br/Brisa. O formulário é simples, aí tá, você consegue preencher, fora a carta de motivação, você tem que pensar um pouquinho. Mas o resto enfim, é fácil de preencher. A gente deve estar aberto até dia 11 de março, que é segunda-feira. Não sei se a gente vai prorrogar isso, e talvez não. Então, tentem ser pontuais. E os projetos, boa pergunta aí, tal, Guilherme. Não, os projetos não têm áreas pré-definidas. O que a gente fez ano retrasado e que a gente gostaria de fazer agora foi abrir no final do curso ali, tal, abrir um pitch público para projetos, assim, cara, propõe seu projeto aqui e tal, mande, deixou uma planilha para as pessoas mandarem ali e tal, estimativa de tempo, de número de artigos, etc e tal, que são coisas básicas de factibilidade. A gente deve ter recebido, na época, uns 8 projetos, talvez, não sei. O que eu me lembro é mais ou menos isso.
Acho que é isso, 8 novos
A
projetos.
gente teve um round de discutir cada um deles, e aí a comissão, enfim, o pessoal mais experiente do Brisa ali tal, de chegar e ver, pô, até que ponto isso aqui é factível, uma boa ideia, uma má ideia, se vai demorar tempo demais. A gente tem um certo olho, tipo assim, cara, isso aqui vai naufragar porque vai demorar tempo demais, porque a gente já cometeu esse erro algumas vezes. Então, assim, acho que a gente teve esse round de discutir factibilidade com cada autor de projeto. Algumas pessoas, inclusive, cara, eu retiro o meu, vou aderir esse outro aqui e tal. E acho que faz parte dessa decisão colaborativa. Eu não me lembro se a gente naturalmente evoluiu para ter 4 ou 5 projetos, ou se a gente chegou a fazer uma votação. A primeira vez a gente fez uma votação, né? Eu acho que a segunda vez talvez nem tenha tido, assim, acho que nas
Acho que
discussões...
foi meio natural, alguns foram sendo
Mas a
gerados.
ideia é desse processo meio iterativo, assim, de receber ideias, e aí as ideias estão abertas para qualquer coisa e tal. E acho que é legal que as pessoas tragam suas ideias e tentar avaliar o que vale a pena fazer, né? Obviamente a gente tem mais ideias do que a gente consegue desenvolver, mas, assim, acho que é natural, a gente conseguiu selecionar o que parecia mais factível ali e tal, e encaixar as pessoas que estavam a fim de trabalhar nas coisas nos diferentes projetos. A gente começou com 5, aí um morreu. A gente tem 4 dos que a gente começou em 22, estão ativos ainda, né? Porque morreu, tinha pouca gente, distribuindo nos outros projetos. Alguém mais? Bom, senão acho que muito obrigado aí, tá, em nome da comissão organizadora do Brisa, e, enfim, muito bom ter sala cheia aqui, né? A gente teve lotado uma parte do simpósio, assim, e tal, que não é tão fácil de fazer em 2024. E, enfim, estamos aí, esperamos que a gente possa ver alguns de vocês no curso e no cotidiano do Brisa aí, tal, nos próximos meses aí, tal. A gente não vai conseguir aceitar todo mundo, a gente já tem mais inscrições do que vagas, mas esperamos que a gente possa trazer um grupo legal de gente para renovar os nossos quadros aí também.
Obrigada,
Alguém
gente.
mais? Então tá. Tchau, gente. Foi um prazer aí. Mário, você tem que desligar o streaming e parar a gravação aqui.